Well, hello, class. We are not finishing, almost. We're continuing our path down, though. So, the last lecture talked a lot about digestion and the enzymes involved, and we'll hit that again. And now we'll look at absorption in the intestines, and we'll keep going. Nice tattoo, by the way. The liver. So, the liver is your largest organ inside your body. I mean, you know I taught you the skin is the largest organ. But this thing is found up here, the upper right quadrant. It's huge. It's going to be protected by your rib cage here, and it can come out a little bit like that. If you have hepatitis, something like that, they can actually they'll feel, see if they can feel your liver, see if it's enlarged. It looks like that, raw liver. Liver and onions is delicious. You either love it or you hate it. But liver, when you look at nutrition wise, you look at, how do I get vitamin A or K, all these things. Liver is always up there, Then you always look for what else can I eat? I don't want to eat liver. But it fills a lot of nutrients, vitamins, because you store a lot in your liver. And wait till you see, your liver just does so many functions. You can't live long without a liver, as you'll see. Yeah, you can see liver, a typical person weighs like 3.2 pounds. Yeah, you can see that. Your brain's about 3 pounds. Interesting. All right, so one thing about the liver, want you guys to remember, is that it gets all of the blood that drains from your organs where you're going to absorb things. So you don't want to eat a bunch of alcohol and drugs and tons of Halloween candy and have all that absorptive material go right into your inferior vena cava and out to your brain. So your liver is going to take care of things in your bloodstream from what you're eating before it takes that blood and filters it and gives it to your body. So this huge organ is going to take blood, as you can see, from your stomach. I'll make it blue. From your stomach, your spleen, also, where you break down blood cells. You want that to go to your liver too. And all of your small intestine. And then it's going to come in your large intestine also from a couple different veins. But all these veins that drain these organs of absorption in your spleen go into your liver in the hepatic portal vein. Indeed. And then all the capillaries in your liver can take care of the blood, anything you absorb. A lot of bacteria too. A good chance of it getting in, especially your large intestine, into your small intestine. So your liver can filter it as well before it returns that blood through hepatic veins into your inferior vena cava just before your heart. Well, so some anatomy, some liver anatomy. It has 4 lobes, your liver. It's got the biggest is the right lobe, and then the left lobe separated by this ligament. There's even a ligament here, this round ligament that goes down to your belly button. And that used to be, in the fetus, used to be umbilical vein would drain. You'd get blood from your placenta from mom, and that would go right into your liver and right into your heart. So your lungs wouldn't get the oxygen, you'd get it from mom, and it'd go right up this thing. And as soon as you're born, that what used to be a big old vein is just going to turn into a ligament. Just like, remember that bypass in our heart, that arterial ligament that would bypass the lungs, that turns from a duct to a ligament. Same thing here. It's going to turn from umbilical vein to this ligament. It's going to come up from your belly button to the liver. Okay. The top of the liver, there's an area up here that it's going to be right up on the diaphragm right there. Remember, your kidney on that side is pushed down a little because the liver is so big. Then underneath the liver, there's the other 2 lobes. So right and left are obvious looking at, from the front. Then it's going to be the quadrate, because it's kind of square like a quadrangle between the gallbladder and that ligament. And then caudate. It's kind of hidden there, but caudate. And caudate means tail. So it's on the back, kind of square. And these are not as clearly defined as looking from the right as right or left, but those are the 4 lobes of the liver. And you can live without a lot of the liver. You can donate a piece of your liver and be just fine, but you don't live long without some enough liver. And look at underside, you can see your gallbladder. It's stuck to the underside, just peeking out here. But it is, in fact, bright green, kind of a forest green. Even cadavers, you can sometimes see on their skin a green stain on the skin. After you die, that bile will leak out. So it is brightly green colored, the bile inside of the gallbladder. And the gallbladder stores bile, concentrates it. And then you need to know this duct system here at the gallbladder. I think I'll tell you right now. So here's going to be the gallbladder right here. And then there's going to be ducts that come in from the liver. Yeah, it's going to make a Y. You guys see that Y? And the ducts from the that are making the bile from the liver are going to be called hepatic ducts. Anything hepatic means liver. So that's where the bile is produced. In the liver, it's going to go down these hepatic ducts. And then from the gallbladder, we call that the cystic duct. Cystic means bladder. And so hepatic and cystic ducts come together, and then we call this the bile duct, or common bile duct. And that's going to carry it down. So in normal functioning, your liver is always making bile, and the bile is going to come down. It goes all the way down to the end of this duct, and the sphincter, the hepatopancreatic sphincter, is going to be closed. So the bile can't go out there, so it's going to back up, come up the cystic duct, and it's going to fill up the gallbladder. So between meals, your gallbladder fills with bile, and it's actually the water's taken out, it concentrates it. And then when you have a fatty meal, do you guys remember the hormones? CCK, cholecystokinin, that hormone is going to cause the gallbladder, there's muscle on it, to contract. It's going to open up the sphincter, and that juice, the bile, will be squirted out along with the pancreatic juice. All right. So your gallbladder, you can see, stuck to the other side of the liver. You can have the gallbladder removed, very common surgery, and then bile simply, it's stored in the duct a little bit, not stored as efficiently, but you can live a normal life. And then I see coming into this little portal into the liver, three things. I see the big hepatic portal vein, that blue one, that's carrying all the blood from your stomach and intestines. It's going to go into the liver for processing. Then I see an artery. What's the name of the artery? Hepatic artery. It's going to the liver. And then I see the bile duct going the opposite direction, taking bile out. So the artery and vein take blood into the liver, and the bile duct is taking bile away from the liver to go to the intestine. Right? You can see where the liver sits, looking at that. It's going to be upper right. It goes all the way across, really, but most of it's in the upper right, like that. And here, again, you've seen a little remnant of fetal circulation is going to be that big umbilical vein, which is going to take blood from your placenta right through the liver. The other two umbilical arteries, they also, as soon as you're born, they turn to ligaments. They come from either side too. They make up the umbilical vein. All right, taking a look at liver histology. When you look at a liver, it's very characteristic looking at it, and you see kind of these lobes. They're kind of these polygons like that. And what you see in the middle is this hole like that. That middle is going to be that central vein. And we're going to see the idea here is that at each of these corners, it's kind of a stop sign shaped, you're going to have blood from both hepatic portal, and fresh blood is going to come in, and it's going to percolate down to these central veins. And on its way percolating in, the blood can be worked on. You can get rid of the alcohol. You can store the sugar. You can make blood proteins and put it into the blood. So these liver cells, we call liver cells hepatocytes. Hepatic is liver. Hepatocytes are liver cells. They fill your liver, and they're very active working at modifying your blood, making sure the sugar is right, making all the blood proteins, detoxifying things, filtering out bacteria. So these liver cells, very important, hepatocytes, in that they work on the blood. And they don't actually filter out bacteria. You're going to see you have white blood cells that do, but the liver itself will. Now, I show pig liver there because honestly, that's what we often look at in histology classes, because you can see the lobules better. Humans don't have as many of this like connective tissue here, so kind of you don't see it as easily, that kind of structure. You can see a central vein here, but it's kind of hard to picture the lobule. A little histology note. So this shows you kind of how we think the liver works. And again, it's really a hypothesis, because there's other ideas I learned too about how this may work. But this is the main paradigm, the idea that we go by. And at each one of these corners, you're going to have a triad. You're going to have three things. And the biggest is your hepatic portal vein. And that's taking blood from where you absorb nutrients and everything from your gut. And then you get fresh blood from the hepatic artery. So it's high in oxygen, it's fresh. You need that too. If you just got portal vein blood, your liver wouldn't survive because it won't get enough fresh blood. So it's about a 70-30 mix of portal vein blood, which is low in oxygen but high in nutrients, everything you just absorbed, and fresh blood, the hepatic artery blood, which is going to be high in oxygen. Yeah. And there's even diseases where you see the cells on the outside are going to get lots of oxygen. As you get closer to the center, they get less and less. Also with toxins too, sometimes things affect the outer part of the lobe, because that gets the first brunt of the blood that you're absorbing, and they get less as you get towards the inside. It's more and more filtered. And then the third thing with this triad in green always is a bile duct, and that is going the opposite direction. Blood is percolating towards the inside, and the hepatocytes make the bile, and that moves towards the outside, and it's collecting these little bile tiny ductules that go to bigger ones, to eventually big hepatic ducts will leave the liver with all the bile that was made by these liver cells. Yeah. So another view too. So you kind of see this shape here. And at the corners, you're going to have this triad always. Three things: hepatic portal vein, hepatic artery, bile duct. And bile is moving outward, and the blood is always percolating inward, like that. And the bile is made in the liver cells, hepatocytes, and little caniculi, little canals carry it out to bile ducts that eventually will all go, and it's going to be stored in your gallbladder for digestion. The blood, it percolates in, as I mentioned, and it goes through these little sinusoids, like these little sinusoids, like a sinus is a space, right? So these little capillaries that kind of drain the blood inward, and it makes it go through a complex sponge kind of arrangement. So the blood doesn't just quickly, like a waterfall, pour into that central vein in the middle. It's made to slow down and bump against these hepatocytes. So these little sinusoids are lined by hepatic cells that are able to work on the blood. Ah, beautiful picture here, yeah. So you guys see, you get about 70% old blood coming out of the hepatic portal vein. 30% of blood is nice and fresh. It comes right from your aorta. And then in green, you see these cells are always making bile. It goes out these little canals, these little ducts. That will be bile is going to leave the liver. It's a product of it. And blood goes through it. And all the blood has to go past these hepatocytes into this central vein in the middle. All the central veins come together to eventually make a hepatic vein that's going to drain the liver back into the inferior vena cava. And the immune cells, these are just macrophages. We call them Kupffer cells when they're in the liver. And they will just hang out in the sinusoids and they'll eat the bad guys. So if you got a lot of bacteria that's kind of made its way into the bloodstream from your gut, Kupffer cells in your liver will take care of them so that the blood going to your heart doesn't have all that debris or pathogens. Maybe. So if someone asks you, what does your liver do? Some of you know, well, it takes care of my alcohol. It's going to take care of it, and it does. Alcohol is broken down into some harmless byproducts, and it's fine if you drink a little bit, but you overwhelm your liver. If you keep adding alcohol, there's only so much enzyme, and so then alcohol levels rise in your bloodstream because your liver can't take care of it. But really, sugars. I mean, when you guys eat a bunch of candy, all that sugar goes to your liver where it's converted to glycogen and stored in your liver. Then between meals, under the influence of glucagon hormone, that glycogen is going to be broken down into glucose, and it keeps your blood sugar pretty steady. So your liver stores a lot of the sugar, will store it or release it. Can also make sugar from proteins, called gluconeogenesis, where it's going to get, let's say you're not eating any carbs since you're just on an Atkins diet, just eating protein, and that's going to make energy from that as well. It can. So definitely glucose metabolism stores and it releases sugar when it needs to. Your liver makes all of your proteins in your blood, albumins, fibrinogen for clotting, all these things. Your liver makes those. So if you have liver failure, you often have protein problems. You're going to have clotting problems, and you're going to have edema because you're not making the proteins that are necessary for that osmosis in your blood, right? So it makes all these proteins. It's a factory that can make all these things. Yeah, it's going to, when it breaks down proteins, it's going to deaminate, make ammonia, and that's going to be, it's going to make urea. So it's going to make the urea that will travel in your blood that your kidneys will filter out, and you get rid of nitrogenous products that way. But all your plasma proteins, damn, it can make anything. Yeah, it builds things from these amino acids. It can build any kind of proteins that you need. And it can convert amino acids from one form to another. You're going to see, except for some that are called essential, you know, there's amino acids that you need to get from your diet. It can't make those, but it can make all the what's called non-essential, meaning your liver can make whatever you need. So pretty awesome. It's filled with, that's why it's so nutritious. It has so many vitamins. It stores vitamin A, vitamin D. It's going to store iron big time. It takes iron and stores it as ferritin, the substance, so that when you need iron, it can release it. Your liver filters your blood, definitely. Like I said, all those Kupffer cells will be there. Those macrophages will be filtering it. With your spleen, it helps destroy aged red blood cells, mostly your spleen. So the blood cells, remember, they only last about 120 days. They start wearing out and your spleen destroys them. Your liver can do that too. And remember, people with a splenectomy, your liver has to take over all the functions of your spleen if you don't have a spleen anymore, if it's removed. So, and even we talked about hemopoiesis, when you make red blood cells, your liver used to do it for you before your bone marrow takes over. So your liver can take up that function a little bit, can make red blood cells in emergency cases. And it definitely takes your blood, and it's going to help detoxify poisons like alcohol. Whew. So your liver, you can see, you don't live long once your liver starts failing. Because all these important things, the proteins, what filters your blood, it's all these things your liver does. Yeah, this is a beautiful chart that kind of summarizes. Oh, just as an aside, eating liver, I said how nutritious it is, but apparently polar bear liver, if you eat that, you will die. I know. Because it has so much vitamin A, it can cause hypervitaminosis A. So why the hell does a polar bear have so much vitamin A? Well, they eat a lot of seals, and seals, vitamin A is used they have very rapid growth, and so we believe it just concentrates it. Yeah. So fun fact. Okay, so liver cancer, often it's spread from colon cancer, but it could start in liver cells, and this shows these tumors. And what you will see, it's going to often rapidly grows, is that the liver gets taken over by cancer. And you all should know what jaundice is. Jaundice is a yellowing of the skin. You see it in the whites of the eyes. And it's a buildup of those bilirubin and biliverdin. If your liver is failing, that bile backs up and goes into the bloodstream, and you start seeing this yellowish. Cirrhosis is, you hear about it as someone drinks too much, it gets cirrhosis of the liver, but that's one cause. But cirrhosis is when you take normal, healthy liver tissue and you turn it into scar tissue, connective tissue. And so more and more the liver becomes useless for its normal functioning. So with cancer or cirrhosis, you have less and less liver function, and you'll see problems with glucose levels, or especially blood proteins are not right. You can see it's called portal hypertension. Remember, all that blood goes to the liver. Well, if the liver is blocked, you have this backup of blood that's trying to leave all of your guts, and it can't. And so blood pressure rises behind it. You can have edema. Sometimes blood vessels in your skin around your belly button will start, the veins will start engorging because the blood will find a different way to get back. So portal hypertension is high blood pressure because your liver is blocking the blood, all that blood that should get through it. And cirrhosis, if you continually assault your liver with drinking or drugs, it kills the liver cells and turns them into connective tissue, and more the liver gets taken up. You hear fatty liver. You can get a transplant. Of course, a lot of people on a waiting list, but you can take a piece of someone else's liver, and the donor can live with losing some liver. It's okay. It will regenerate some, and you don't need a whole liver. And the recipient, as long as it takes, can take over that liver function, and you can save a life. And they have artificial, like a dialysis machine for kidneys. You can get on a machine that takes over the liver function somewhat. It's kind of a bridge between liver failing and getting a new liver. Hepatitis. Remember, hepatic, everything liver. Hepatitis is your liver inflamed, and it can be for some other reasons, but it's almost always viral, an infection of the liver because of these viruses. And it's amazingly common. Look at that. 1 in 12 people worldwide. So often hepatitis, you notice you feel sick and then you get better, but you can have some forms that are really nasty that can kill you. I do want you to know hepatitis A, B, and C. Type A, you get from feces, like if you go somewhere and eat a lettuce, a salad that hasn't been washed, that kind of thing. The mildest is hepatitis A. And if you travel some places around the world with less sanitation, you can take some drugs before you go. Hep B, you can get vaccine for that. And working in a lab in biology, I vaccinated a long time ago. But B is usually from blood or body fluids, so unprotected sex or blood transfusion or something like that. And so it's a little harder to get than type A. You can just eat dirty vegetables. And Hep C also is transmitted by blood, needles, or sex, often the way you can get Hep C. And there's new drugs for all of these too. I used to be Hep C. There was nothing you can do, but in the last 5 years or so, they've come up with things to combat the virus. See, Hep C, you can see some places where you get it. So IV drug, if you share needles, it's taking the virus from one person to another. Sex, transfusions, you get it in the blood supply. And of course, some they just don't know exactly where you got it from. So hepatitis, viral. Again, we have some vaccines to protect against it. And there's actually A, B, C, there's D, E, there's a whole bunch of other ones too, but A, B, and C are the common ones. All right, let's talk bile. Bile is a thick, yellowish, greenish fluid that is going to be stored in your gallbladder. Remember, it's made in your liver, stored in your gallbladder. And unlike your urinary bladder just holds pee, urine, your gallbladder actually is going to concentrate it, will remove water. And so it's this thick fluid. And it's going to squirt out into your duodenum, the first part of your small intestine, and it's going to hit that food that's coming from your stomach. And it really works on fats. What's called emulsify fats. So it takes a glob of fat and it makes it into little globs of fat. And that is going to allow the enzymes to be able to work on it because it has more surface area. So emulsifying fats is like a detergent or a soap. The bile salts are going to do that. It's going to have an area that is hydrophobic and hydrophilic. And so it's going to surround the fats, so the hydrophobic part, and the hydrophilic part is going to face the water, and it's going to turn into little pieces for more surface area so that the lipases can break down the fat. So bile salts emulsify fats, which is what liquid detergent does when you wash dishes. It's going to take the fat and make it into little pieces instead of being it just like glomming up. If you don't have enough bile, you could have a deficiency of some fat-soluble vitamins, because when you absorb fats, some of those vitamins, like K, needs to be brought in that way. The color of it comes from that bilirubin, biliverdin. The bile pigments come from breakdown of red blood cells. And that's what causes jaundice. If you can't get rid of it in your bile, it's going to build up in your blood, and your whites of your eyes look yellow. And so in bile, the bile salts are doing the only part that does the digestion, really. The pigments are just leaving, and cholesterol, which is used to make the salts, is also gotten rid of too. That's another way you can get rid of that. And yeah, some electrolytes too. But this is what makes up the bile. And the salt's doing the work, and it's a way you get rid of some cholesterol, actually get rid of it. And these bile salts, remember, they're squirted from the bile into your intestines. Then a lot of it gets recycled. Because remember, the absorption from your gut goes back to the liver. And so it can be used to make more bile, right? And some of it always goes out in your waste, so you have to make new bile salts. But a lot of it is just recycled back and forth. Yeah, so there's that gallbladder. Yeah, it's a little bladder, it's stuck underneath your liver. And remember the ducts? The duct leaving is the cystic duct. Hepatic ducts drain the liver, and then the bile duct goes all the way down to enter your intestine, that little papilla. We talked gallstones, of course. We talked about kidney stones already. Gallstones are usually made of cholesterol, and if you have too high cholesterol, or some of it's genetic, some we just don't know why some people get gallstones and others don't. You know, obesity and a lot of things can contribute to it, but you have this thick fluid, and if you start getting precipitate, concretions will form these stones. Just like in the kidney, we have a little bit of crystals that will form in there. Once they start from concentrated urine, they grow. Cool, some are pigment stones. That's kind of interesting too. And these gallstones, you'll have pain in that upper right quadrant, sometimes intense pain. And they've got options. They can try to break them up from the outside, little shock waves. They can do some surgery. They can put in a little tiny, like a little tool that's a little grasper and kind of grasp the stone. Or they can do a gallbladder removal. And they can do it now just with small little incisions and laparoscopically take it out. And people say, well, how can you live without a gallbladder? Well, you can. You make bile, and it's not stored in a gallbladder. It's just kind of stored in the duct and released. If it's too much, it just gets reabsorbed by your body. And I always thought, well, real problem with fatty foods, right? Because you need bile to digest the fats. And it's true. You need to, people that have gallbladder removed will get some counseling on maybe not eating large fatty meals because it's more difficult. But I've talked to people with gallbladder removed, and I just eat a normal diet, so it's not a huge deal. So bile release happens, like I said, after fatty meals. It's most important because those bile salts, that's how you're going to emulsify the fats and be able to have the lipases work on them. So that when that acidic chyme hits the duodenum, it's going to stretch the duodenum, the acid's going to stimulate, and you're going to release that cholecystokinin, the CCK. It's released by cells, intestinal cells make it, release it in the bloodstream, and it's going to, when it hits the gallbladder, it's going to make the smooth muscle in the gallbladder contract, and it's going to squeeze that gallbladder, and the bile in it's going to go down that tube, and that sphincter, hepatic pancreatic sphincter will relax and it will squirt the bile along with pancreatic juice on that chyme that's coming from the stomach. Great, so these hormones, okay, questions on the test. You should know what gastrin does, what cholecystokinin and secretin do. Yeah, this shows you the bile salts are doing their thing. I've already talked about this, but here it is in words. You can see a fat globule just surrounded by bile salts to help break it down. All right. So the small intestine, I've been throwing around duodenum quite a bit. Now I'm going to actually define the three parts to the small intestine. And it's small because of its diameter. The large intestine is large because it's wider. Small intestine is actually the longest part of your whole alimentary canal. It says 7 meters here, here 6 meters. It depends on the person. Depends on whether you're alive or dead too. The muscle tone changes. Look at the size of it, but it's clearly super long. And in lab, I had this tube that's marked off to just impress upon you how long this is. 7-8 meters. You realize how long that is? That's really long. But it's all coiled up and inside of your abdomen down here. So what happens in the small intestine? The main function, if you remember nothing else, is absorption. It is going to absorb the nutrients and the water and the vitamins from your food. A lot of digestion too. It's hit with all those enzymes from your pancreas, right? So you're going to digest sugars and proteins and fats. It's going to continue to chemically digest your food. By the time it hits it there, it's like milkshake, watery consistency, right? You brought it in as carrots and peanuts and whatever you're eating, right? And you're going to chew that up, mix it with water, hit it with acid, churn it, and turn it into this like milkshake so that it's has a lot of surface area, so you can, by the end of your small intestine, you've taken out all the good stuff and you just have waste at the other end. So it's impressive. It's going to move slowly down the small intestine. It's going to be segmentation, it's going to churn it up. Peristalsis is going to slowly move it along until by the end of your small intestine, it's just going to be that indigestible waste and water that you want to get rid of. Well, you want to take the water back, but get rid of the waste. So the anatomy-wise, the first about foot of it's called the duodenum. Don't say duodenum. Duodenum. Then the next of it, you can't really tell the next two parts apart just looking at it easily. There's some subtle clues, but jejunum and ileum make up 2/5 and then the last 3/5. How they get the words, a duodenum like Deuteronomy in the Bible, you know, looking at, it was about 12 finger breaths is about how long it was. Jejune, that's a word for you liberal arts people to use. Jejune means I'm feeling jejune, I'm feeling empty, you know. And in death, that part of the intestine would often be emptied out because the muscles would push it out. It'd be empty at autopsy. So jejune means feeling empty. And then ileum means twisted, kind of moves around like that. And as you move your way down, so the duodenum would be, like I say, only about a foot, and then there'll be ileum. I'm just straightening it out, and then finally, I'm sorry, that's the jejunum, and then finally the ileum. And it actually gets progressively smaller. So if you guys swallow something like a marble, it often will get caught at the end of the ileum. It'll make it through the stomach, through the pyloric sphincter, through the jejunum, but the ileum is where it gets the narrowest, kind of as an aside. This thing's, well, it might be the bottleneck right there. And by the time it gets to large intestine, it's got plenty of space. So that's kind of a small area. So your small intestines, they make up all, when you cut open your guts, they spill out. They're all these like worm-like guts. And duodenum is stuck to the back wall. It's actually retroperitoneal. It's stuck. It's not very mobile. And then at the end of that like C shape, that first little curve of the duodenum, then it kind of is all free inside your abdominal cavity. You can move them all around. Looking in the small intestine, it looks like velvet. It looks like velvet in there. And you'll see food material in there too, but this velvety is because of all these little fingers, just like a like a plush towel, has all these little fingers of fabric that make more surface area. The surface area, 600 square meters. That just blows my mind. I mean, the size of the baseball diamond, the infield. That's just it's a lot of surface area that we have as mammals because we want to get every calorie out because we use so much, keeping our high metabolism. And so for the surface area, when you look at the beginning of the small intestine, these plica or these folds will increase surface area. And then the villi are the fingers that come out. And then on the cells on the villi are the microvilli. So you have these successive layers that increase the surface area again and again and again for absorption. And absorption is how we take things that we eat and bring them into our bloodstream so that we can use them for energy and for building our bodies, everything we need. So in this slurry, this milkshakey fluid, where the enzymes are going to break down all the carbs into simple sugars, all the proteins into amino acids, the triglycerides got to turn into just the individual fatty acids. Those have got to be brought into these columnar cells that line our intestines. Often it's active transport. We're using energy. Sometimes it's facilitated diffusion. In cases of water, it's osmosis, just being brought in. But all these things are brought into these cells that line the whole gut tube. And you got to keep out the bad guys, keep out the bacteria, but you want to let in what you want to let in. Once they're in the cells, they could be modified, but then they're kicked out the other side, and all these capillary beds will absorb those nutrients and carry it to the liver and eventually to your body. Here's a view so you can see. The duodenum is just right here, just this first part, and it's stuck. It's just stuck back there. And then the rest of them, the jejunum and ileum, I mean, you can't tell really where they separate. There's some gross anatomy differences where the fat comes in, and if you look at histologically, the ileum has more bacteria at the end part, and it's going to have more lymphatic tissue, but it's really kind of subtle. But we talk about jejunum and ileum being the rest of the small intestine. And the ileum will empty into the large intestine. Yeah, everyone's a little different, but you're looking at here, they put some dyes, you can see that. So small intestine makes up all this in the middle, and you see the large intestine will come around like this. I already told you guys about this, but part of the peritoneum in here that is going to hang over the intestines is this greater omentum, and it's a double layered membrane with tons of fat in it. So that's the greater omentum. The first thing you see when you open up the guts, you got to pull that back and you'll be able to see the intestines. Looking at the villi. So we're looking microscopically now, and each little finger like that, you see it has blood vessels in each little finger to absorb the sugars and amino acids. And then don't forget, the lymphatic is going to come in a lymphatic vessel, comes in each one of these little fingers, and that's how you're going to carry the fats away. And those lymphatics are called lacteals, like lactating milk. They turn kind of milky because all the fat's going to be absorbed in that after a fatty meal. And these cells on the surface, always they're being assaulted by all these enzymes. They're going to break them down. So cell turnover, it says on here, about every 6 days. Yeah, all the cells are digested and you make new ones. So high mitotic activity, lots of cell division. It says here a quarter of the bulk of the feces is actually just your own intestinal cells that are making it down. Yeah. So these intestinal cells, highly active, and they've got microvilli. Now the microvilli don't move, they're not cilia, they don't wave, but they just make more surface area. And the cells surrounding them are columnar, again, highly active. They're pumping in nutrients, and they're might do some little work on them before releasing them to go to your bloodstream. So the small intestine itself is, of course, has enzymes on it. Like I talked about a lot of enzymes that came out of that pancreatic juice, right? But on the surface of the intestines, you've got to have that enterokinase, for instance. You got to have that because that breaks the trypsinogen into trypsin to make it active to break down proteins. And then the cells of the small intestine are also going to make some hormones. Remember, it gets hit by chyme and then it releases secretin into the bloodstream. Secretin makes your pancreas go. And then CCK is going to do the same thing. It also involves the gallbladder too, makes that kind of excited. Yeah. And then the intestines are going to send signals back to the stomach to tell it to slow down. That's a somatostatin is going to inhibit it. And then on the surface of the small intestine is also enzymes to break down proteins, pepsidases. You can see sucrase breaks down sucrose, maltose, lactase. And if you're lactose intolerant, you don't make enough lactase. And so milk sugars, they end up just not being digested. Bacteria work on it, making gas, and all of the excess sugars will make more water by osmosis remain in the waste. Yep. And some lipases, again, to break down fats. And the whole small intestine makes lots of mucus, right? It's got goblet cells throughout it. And in the very beginning, there's big mucus glands that make a bunch of mucus because once that acidic chyme leaves the stomach, your intestines need to be protected. So hormones, enzymes, and mucus all being produced by the small intestine. Yeah, the small intestine, it's got to protect itself with mucus, right? And once it gets hit by that food that's leaving the stomach, your small intestine has got to have that enterogastric reflex. It's got to go back and tell the stomach, okay, dude, we're getting all this stomach contents. Now you need to slow down and just give us a little bit at a time. Nice big chart here looking at all the different enzymes. So make sure you study this, like what happens in your saliva, that amylase. So starches begin digestion in your mouth. Your stomach, what happens there? Pepsin is the main thing. It's going to break down proteins and a little bit of lipase too. Your small intestine, everything, right? Your pancreatic juice has everything. Your intestine is secreting enzymes. That's where you finish the chemical digestion. You break down everything so you can absorb it in that small intestine. And then there's hormones too, given off by the stomach, like gastrin, by the intestines, like secretin and CCK. Hormones are given off. And then by the time that food finishes the small intestine, you've taken all the good stuff out. And the large intestine, we're going to see, is just going to reclaim water and electrolytes and just get the waste kind of ready. The absorption of the small intestine, again, surface area is key. Surface area is key. The more surface area, the more you can absorb. And us mammals are good at it, at absorbing everything that's out of our diet. And you can't absorb starches or proteins. They all got to be broken down into amino acids and monosaccharides, little pieces so you can absorb them. And so definitely you're going to pump in the sugars and amino acids. Those are the good stuff right from your diet. Pump those in. You got to break down large proteins. Now we'll talk about fats. Fats are a pain in the ass. They are don't dissolve in water. So just carrying it in our blood requires carrier proteins, right? And then bringing them into our absorption is also a pain too. You saw you needed bile salts just to break into small pieces. Then I'll talk about how those small pieces can be absorbed and then how we prepare them to go through the lymph system back to our blood. Water, electrolytes, a lot of that is osmosis. Once you start pumping all the sugar and amino acids into the bloodstream, it makes the remaining fluid more watery. And so that's just going to move, and the salts are going to move into the bloodstream just passively. So looking at some of this digestion, you can see, remember, God, this was last semester, hydrolysis is how we break apart. Remember that? And so you use water, it's going to come in this bond and break it into its monosaccharides. So whatever sugars you guys are eating, it's milk sugar or sugar from beets or sugar cane or anywhere, even starches that are broken down into the sugars, they're going to be broken down by these enzymes. You got specific ones to break down different kinds of sugars. And so glucose, hell yeah, you can absorb that in the bloodstream. That you can handle. You can't handle bigger things like this. The proteins, you're going to have pepsidases that break down peptide bonds. You put the water in here and you split it apart. And you can absorb individual amino acids. And then fats. Look at this. So for a triglyceride, use 3 waters. And lipases will break those apart into these 3 long fatty chains. And then the glycerol over here. And those you can absorb into the cells. So these are what the enzymes do. They're going to break apart these larger molecules into little smaller pieces that can be absorbed. So here's the deal with fats. Bile salts will make it into smaller pieces, where lipases can break apart a triglyceride into those 3 long chains and then the glycerol part over here, and those are pumped into the cells. And then the cells will take those fatty acids and actually put it back together into fats. And these fats will have this kind of a candy coating around them of proteins that allows them to be dissolved in water. So this protein coat allows the fat in the inside to move through and to move inside your blood. And you need to know we call these chylomicrons. These are the center of fats and an outside of proteins. And that allows them, the fats, to move through the blood and not all clump up. And the chylomicrons that are formed are pushed out and they'll be moved through a lacteal. Remember, all your lymph vessels will eventually drain back into your blood, into your veins, right? And so these chylomicrons can make it out to your adipose cells and your muscles to be brought in, and your liver will really work with them. So the liver, it really does a lot with fats. You guys, I think we'll talk about LDL and HDL. You guys get a lipid panel with like hundreds of kinds of fats, and some are high density, low density, meaning a lot of protein or less protein. And there's the good fat and the bad fats. Oh, not only fats there, but also the LDL, HDL in your blood. So there's a lot to talk about here, but this is how fats are brought in. They're brought in the individual pieces. They're reassembled. They get a protein coat around them so they can dissolve. It's called a chylomicron, and that's going to go through the lymphatic system back to the blood. Awesome. All right. So been through a lot, but we made it through the small intestine. And finally, the last lecture, we're going to go to the end. We'll talk about what happens in this large intestine. You can see your small intestines make up most of the length of your alimentary canal. It's about, like I say, 6, 7, 8 meters. And then your large intestine is only going to be about 1.5 meters or so. So it's much smaller per se, but has a larger diameter. And it has these weird little pouches, doesn't it? So we'll talk more about that.